Yes, when I saw the title that had been proposed, first of all, thank you very much to the organisers for this kind invitation, which represents a kind of challenge, because I'm going to have to try and do what the classic miniskirt is supposed to do, touch on everything while covering nothing. Touching nothing while covering everything, whichever you prefer. So I'm going to try. I didn't like the idea of giving a lecture to physicists, because I don't think I should be talking to physicists. You should all be interested in the essential role of iron in biology. So I'm going to talk from the standpoint of a biochemic. And my talk is very dense. I'm going to start off with a very brief introduction, which is hysterical and historical. It will then become clear that there's some hard chemistry that you need to remember about iron, which is fundamental, because if you don't understand that, then you'll make a big mess of anything you do in iron. Then we'll talk very briefly about the brain and iron, because I was asked to simply remind you that the brain is a complex organ and has a large number of different cell types. So I will just briefly mention those before moving into the classes of iron, the classes of iron proteins. Then we come into the last part of the talk, which is essentially the dramatis personae of iron metabolism and iron homeostasis, and we'll meet all the partners who are involved in this, which will enable you to understand what's being taught in much greater detail in the rest of the week. And I'll end up by talking about the dark side of iron, because iron is, of course, toxic, especially when combined with molecular oxygen. So in the beginning, there was iron. The first principal element in the molten mass that condensed and coalesced to form our planet Earth was in fact mostly iron, 80% iron, with a lot of nickel. And it's the fourth most abundant element in the Earth's crust. Of course, aluminium is more abundant, but aluminium, which has a lot of similarities with iron, is however not used by biology at all. Man is responsible for introducing aluminium pollution into the world, not man, not nature. I just remind you of this very important period here, which we will come back to, which is when the first major pollution event in the world's history began, which is when wretched cyanobacteria started discovering oxygenic photosynthesis, transforming water into 4 protons, 4 electrons, and a dioxygen molecule. And as we will see, that consequence as iron builds up in the atmosphere, meant that iron, which had been freely available, suddenly becomes very unavailable. Civilization in humans is often described in 3 ages: the Stone Age, the Bronze Age, and the Iron Age. Because the Stone Age was quite a long time ago, 2 million years ago, and lots of traces of the Stone Age. Then the Stone Age was succeeded by the Bronze Age, using metals, notably copper and tin, and inventions such as the wheel and the ox-drawn plough. But then, between 1200 and 1000 BC, people discovered that if you could heat iron on a wood fire with small amounts of carbon, you produced something which we now know is steel, and which was much harder than bronze, and therefore you created something which was iron metallurgy grew and grew, and iron implements began to create new patterns of more permanent settlement. But on the other hand, iron was so readily available for weapons that you were then able to start making large tracts of changes in population, movements of people all over the face of the earth for 2,000 years, and it changed the face of certainly Europe and Asia. So for 3,000 years or so, until its replacement by the hardened steel of the processes that were developed in the 19th century, iron was the fundamental material of human civilization. Unfortunately, rust is not a good way to preserve archaeological remains, so we don't have an awful lot of archaeology remaining from the Iron Age. Iron chemistry is quite important, and I want to insist on this because it's not just because of physicists, because believe me, one of the problems that we have as biochemics is to try and convince biologists that chemistry matters, and especially when it concerns iron, because iron does some pretty nasty things. You just stick some iron in an aqueous solution, which of course contains dissolved oxygen. It will simply oxidize, hydrolyze, polymerize, precipitate. That's what's going to happen. So you have to be very careful when you do experiments with iron to pretend that you're using iron 2 instead of iron 3. It's not as easy as it sounds. You can actually get iron 4, and we'll see in some of the examples that I give you of how important iron is in enzyme chemistry, that we have lots of enzymes that generate reactive iron 4 species in the process of activating relatively molecular oxygen. One interesting sidelight here, which we'll see in a moment, is that the importance of is that iron can get down to potentials of about plus 0.6, but it can't get up to the kind of potentials that you need to reduce dioxygen. And that's where copper comes in, and you'll see the connection in a few minutes. It's a hard, iron 3 is a hard acid, prefers carboxylate and phenolate ligands rather than imidazole and thiolate. Iron 2 prefers the imidazole and thiolate ones. And then this means that in fact iron 3 is unique in biology. You can selectively chelate iron 3. I would challenge anyone in this audience to selectively chelate iron 2. There are too many other divalent metal ions around which can easily compete. But iron 3 only has 2 analogues in the whole of the periodic table. These are aluminium, gallium. Gallium is extensively used for medical purposes, but is not a biological element. And aluminium has been used as a pollutant in our society, but which I should add, in my humble opinion, having done quite extensive research on the subject, has no major implication directly in Alzheimer's disease. A lot of that was absolute nonsense because it was based on the fact that we were using aluminium grids that were used for electron microscopy and then showed contamination. Aluminium is one of the nastiest elements to do analytical chemistry, believe me, having done it for a few years. I just passed through the iron-oxygen chemistry, but I want to point this little thing here. When you've got iron 2 in the presence of hydrogen peroxide, you make the hydroxyl radical. And this is the most reactive form of reactive oxygen. The chelate effect, which I mentioned earlier, for physicists this is particularly important because you've probably heard of one. Chelate is derived from the Greek claw, and what happens when you have a chelator is that it wraps itself around. Here is the well-known desferrioxamine, desferal, and when it wraps itself around an iron 3 center, you see you form a beautiful hexacoordinate oxygen liganded ferrioxamine. That's what the chelate effect is, and the chelate effect is very important in metal chemistry. The presence of oxygen means that you oxidize iron rapidly. Iron 2 is very water soluble, iron 3 is poorly water soluble. The water solubility of iron 3 is such that you have a concentration of iron 3 in free solution of about 10 to the minus 18 molar, which means that you could swim a long time in the lake over there before you find a single Fe3 plus ion. So iron 3 is not readily soluble. They have a strong iron salts tend to hydrolyze. This is the process, which is the process of hydrolysis here written down. They simply leave a proton continuously, and then you eventually form ferric oxides, FeOH. And these polynuclear clusters then precipitate in the form of ferric oxide and hydroxide. And finally, this is the toxic part of iron that we'll come back to at the end of the talk. In the presence of hydrogen peroxide, iron can form the hydroxyl radical. This reaction is attributed to James Fenton. In fact, Fenton did not discover this reaction. Fenton discovered that if you took H2O2 and iron 2, you could use it as an oxidant for oxidizing organic compounds. Subsequently, the reaction was rediscovered by Haber and Willstetter, who published the paper in German, so nobody cites it anymore, and you tend to get the more frequently Haber-Weiss publication in 1935, which states the obvious thermodynamic impossibility, because the Haber-Weiss reaction says that oxygen plus hydrogen peroxide gives superoxide and hydroxyl radical into which is absolutely impossible in the absence of iron, trace amounts of iron. Anyway, we're not going to rewrite chemical history. Let's continue with this little comment here. So when oxygen started to appear down here, what happened was that iron simply slowly began to precipitate. And what we can see is that in the early stages of evolution, you had high concentrations of iron 2 readily available. Supramolecular oxygen appeared about 10 to the 9 years ago, you could then start seeing iron hydrolyzing, oxidizing, precipitating. And if you look in the Precambrian deposits geologically, you see the red ferric oxide laid down in the geological strata, which simply represents the disappearance of iron from an available form to a totally insoluble form. I'm not going to go into this in detail, but this means that microorganisms have developed strong iron chelators that we call siderophores, which go out and pull iron-3 out of solution, even from the surface of iron minerals. This is how microbes survive, enable themselves to get iron. But the important point I want to make is that since iron-3 was less bioavailable, the curiosity is that copper-2, the oxidized form of copper, is much more bioavailable. Copper-2 salts are much more soluble than copper-1 salts. So copper became available, and that was fortunate because, as we'll see, when it comes to reducing dioxygen, we need to use iron and copper, not iron alone. Iron is an almost essential element. Why do I say an almost essential element? Because it's present, it's required for almost all living organisms, with a small number of microbes that don't use iron. Now, if I was to tell you that milk is a very important source of nutrients for human infants, you would not be surprised. If I was to tell you that milk is very good at preventing itself being used as a source of iron for microbial infection, this might give you a clue as to what kind of microorganism might survive without iron. Lactobacilli. Lactobacilli have developed, in fact, the most important enzyme in iron chemistry is the ribonucleotide reductases, which convert ribonucleotides to deoxyribonucleotides, essential for making DNA. You can't make DNA, you can't make an organism that can reproduce itself. But whereas most of the all of the class 1 and class 3 enzymes are in fact iron-based, the class 2 enzymes are in fact cobalamin-based, so they're cobalt-based. They all generate a thiol radical. This is the, the way in which they do it. A di-iron center and a tyrosine radical, a manganese center and tyrosine radical, and then a few prokaryotes, a manganese iron center and tyrosine. Class 2 is the cobalamin, class 3 is adenosine and an iron-sulfur cluster. They all make this glycol radical, and this enables you to do this reaction, which involves taking a ribonucleotide, the deoxyribonucleotide, using this thiol radical here, and then you use a disulfide, a double dithiol to equip that with that. Interesting, and this is for the physicists, because I thought you liked this kind of thing. It's quite amusing to state that the actual, in the human ribonucleotide reductase, the di-iron center is located down here. The active site when you generate the thiol radical is on the other subunit, colored here in pink, up there. The distance between them is 35 angstroms, and that electron has to hop that distance, and it's believed that it does this through a channel that's made up of a series of aromatic residues. And this radical transfer hopping pathway is quite widely found nowadays in biological oxidation systems. Just a word about the brain. It's 2% of your body mass. It receives 15% of your cardiac output, consumes 20% of your total oxygen consumption, and accounts for 25% of body glucose utilization. And yet, so it's kind of rather gourmand. In fact, it becomes gourmand to the extent that if you drop the glucose levels too quickly, people will simply fall unconscious because they need the time to switch from metabolizing glucose to metabolizing fats. And of course, this is one of the benefits of going on one of these wonderful diets where they cut you down to 400 or 500 calories a day. And you then, of course, start burning your fat. Once you start burning fat, anybody who knows biochemistry knows that you make so-called ketone bodies. One of these ketone bodies is acetone. Acetone is hallucinogenic, so no wonder you come out of the spa feeling $1 million because you're on a high. I shan't go into that in any further detail. It's an interesting sideline. Now, I was asked just to mention brain. I'm not going to, I say, it involves a wide number of processes: neurotransmitter synthesis, myelination of nerves, motor neurons. So what do we know about brain iron metabolism and homeostasis? Well, that's what we're here to find out about. Let me just remind you that, however, in the complexity of the human brain, we have not just the neurons, of which we have at least 3 types. The motor neurons, which take messages down to a nerve, to a muscle cell here. We have the sensory neurons, which collect data, feed it back into from a receptor cell back into another terminal which transmits it on. And then we have interneurons. We also have, of course, different kinds of microglial cells, in addition to the neurons which are myelinated, around which we find the oligodendrocytes going down the myelin sheath. We have astrocytes, we have microglia, and of course recently it's become quite clear that when you look at the blood-brain barrier around the blood endothelial cells, you have these what are now known as neurovascular units. The neurovascular unit consists of the endothelial cells, the pericytes, the astrocytes, the end peak of the astrocytes around the endothelial cell, and then the other cells, microglia and neurons. Typically, one of these arrangements would be found quite frequently all over the brain. So, let's go to iron proteins and their functions. I'm going to classify proteins according to the way the iron is bound. So we can have heme proteins, we can have iron-sulfur proteins, and then we can have proteins that are neither heme nor iron-sulfur. In the first group, we're going to see that we've got oxygen carriers, oxygen activators, and electron transporters. Here's hemoglobin. You know the marvelous story of hemoglobin. Myoglobin, which is a single chain, binds oxygen in a hyperbolic curve. Hemoglobin binds it in a sigmoidal curve. What this means is that when you've got low partial pressure, hemoglobin will release a lot more iron than it does if it was myoglobin. And this is because iron binding is cooperative. What does that mean? It means that the first oxygen atom goes on with great difficulty. The second one goes on better, the third one goes on better, and the affinity for the fourth oxygen is about 100 times that of the first. So, and that's done by transmitting a message between the subunit that gets oxygenated and the helix here, which then transmits to the neighbouring subunit, and so the whole structure is informed. Cuckoo, I've just been oxygenated. Time for you to wake up and grab an oxygen while you can. That's hemoglobin. I like to think of throwing in the odd slide that wakes the audience up, so in case you've fallen asleep, this is a depiction from the walls of Pompeii, showing Mars and Venus obviously in a quite non-intimate relationship. I'm sure he just stopped by for a cup of tea or something. I don't think it was quite like that in Greek mythology, but anyway, Mars was always called the Roman god that represented iron, and copper represented by Venus. And of course you find copper and iron together where? In cytochrome C oxidase, which is the terminal enzyme of the respiratory pathway in your mitochondria. And there we do the brilliant reaction of taking oxygen, 4 electrons and 4 protons, and making 2 molecules of water. Exactly the opposite of what the photosystem 2 in green plants does. But it uses a tetramanganese center rather than copper center. This is an example of the kind of high valence that we get in the peroxidases and the catalases, but we also get it in cytochrome P450. The cytochrome P450s, and this is a slide which everybody should make a careful note of because I'm going to ask you questions on these details at the end of the talk. This is just to give you a kind of brief overview of the kind of reactions that cytochrome P450s can do. Okay, it's enormous. It's got huge capacity to do all sorts of things. we also have cytochromes, and cytochromes are by definition, you saw that hemoglobin has 5 ligands, the 4 ring nitrogens and 1 ligand underneath. Same is true of peroxidase, catalase, cytochrome P450, but the cytochromes have 2 ligands, the protein, and that means that the only thing that a cytochrome can do is flip from iron 2 to iron 3. So they're iron transporters. There is a good theological argument as to whether an electron transporter is an enzyme or is something else, but anyway, they're electron transporters. And we find them all over the place. Here's one example. This is an octaheme cytochrome C. It's a nitrite reductase. One of the 8 hemes only has 5 ligands. The others all act as electron collectors and throw the 6 electrons onto the catalytic side to convert nitrite to ammonia and hydroxide. This is the respiratory chain, and if you want to see just how many cytochromes are involved there, there's none in complex 1 and 2, but there's cytochrome BC1 in complex 3, a cytochrome C that shuttles between here, and then there's a cytochrome C oxidase, which is here inside this. So the mitochondrial respiratory chain contains this interesting Q complex, where the coenzyme Q comes from complexes 1 and 2 and then goes through to complex 3. And that is the structure of the dimeric cytochrome BC1 complex. You can see the 2 hemes, you can see the Q sites, you can see the site where an inhibitor binds, and the iron-sulfur cluster. And now we're going to come to iron-sulfur clusters. But just one last mention. Yes, here's iron-sulfur clusters. So you've got iron-sulfur clusters with 1 iron, with 2 irons, with 3 irons, 4 irons, to 8 irons if you like. Iron-sulfur clusters are not just used for doing electron transfer. The key enzyme of the Krebs cycle, the tricarboxylic acid cycle, which is at the center of metabolism, is the enzyme which converts citrate to isocitrate dehydrogenase, and that is an iron-sulfur protein. But one of the 4 irons in the iron-sulfur cluster is easily exchangeable, and that's the one that the citrate binds to, and in the reaction you change yourself from having citrate, which is a completely symmetrical molecule, into having isocitrate, which is an asymmetrical molecule because the CHOH is on one of the CH2s of the citrate. And this is stereoselective. The enzyme will only put the hydroxyl group on one of the CH2s of citrate, not on the other, because it can bind it at 3 points on the surface of the enzyme. You can do the same thing with alcohol. Alcohol dehydrogenase is also stereoselective, despite the fact that it looks like a symmetrical. Right. This is another quaint enzyme, hydrogenases. These are all the rage at the moment. If there are two protein complexes which are the source of great interest in terms of biotechnology, they are the tetramanganese site of photosystem 2, which converts water to oxygen, protons and electrons, and hydrogenases, which convert protons and electrons to hydrogen. And of course, if you have these two working together, you have the source of making hydrogen and oxygen, and then you could run all sorts of things on something for which the end product is simply water. Right. This is another example of iron-sulfur chemistry. We can make a large amount of highly oxidant species which are involved in carbon-hydrogen bond activation using iron centers, but we cannot just using our heme here. These are the heme types or the non-heme enzyme. You can make all these types of reactions. But you can also make the same kind of chemistry by making adenosine radicals from S-adenosine methionine and a 4 3-iron 4-sulfur cluster. This is the class of enzymes that we call monoheme. These are non-heme, non-iron sulfur proteins, and they contain one single iron atom. They catalyze reactions such as the catechol dioxygenase reaction, the dioxygenases, the alpha ketoglycerate dependent, the pairing dependent hydroxylase, and another collection of them. And these include Among them, the proline and lysyl hydroxylases that are involved in the maturation of collagen, very important reactions, and the phenylalanine, tyrosine, and tryptophan hydroxylases, which are essential for making neurotransmitters like serotonin and adrenaline. This is simply to show you that just a little bit of tweaking of the chemistry here can decide whether you do an extradiol or an intradiol dioxygenase reaction in catechol. And these are the last category, di-iron enzymes. The first thing that's striking is that they all have this tetra. By the way that we tend to depict proteins is that these squiggly things, these are meant to be alpha helices. And if you see something with an arrow and a point on it, it's a beta sheet. So all of these are very similar. Four helix bundle structures. They all have very similar reaction centers, reactive centers, and they catalyze reactions which vary from methane monooxygenase, which transforms methane into methanol, toluene into an alcohol, riboneutraldehyde reductase, which is an iron transporter among things, acyl-CoA desaturase, bacterioferritin, methyl-erythrin. This one puts double bonds into fatty acids, and this one is in fact an oxygen transporter. So you've got all this variety just around a di-iron center. Having convinced you that iron is important in biology, where am I in time? I'm going to have to go like the clappers for the next 15 minutes. Sorry about that. No, I've got to 1:15, haven't I?
Yes.
Oh, I've got plenty to do. Right, I can always tell a couple more jokes. No, sorry. Right. Now, we now come to the to show you the 4 reactions which are involved in cellular iron metabolism. Right? Iron needs to be taken up by a cell. It needs to be utilized by a cell. It needs to be stored by a cell in case it needs it, and it needs to be exported. Right? And all of that is depicted on this slide beautifully and simply. So if you remember one thing from this lecture about iron metabolism, this is the slide you want to remember. The easiest one to remember, which is there's the least amount of garbage around. So iron is taken up by most cells via a mechanism that involves transferrin, the transferrin receptor. It also requires a ferroreductase and an iron dimetal transporter. Storage is the protein that I've spent most of my life working on since 1968. It's called ferritin. Iron proteins of the various sorts that we just saw in the previous section are the utilization end of it, and then there's a protein called ferroportin. Now here is a word of warning to the young people among you. When you've got a good piece of information, don't go to a meeting and talk about it. Put it in print first. Okay. Our colleague Sandy Mackay at King's College London discovered an iron enzyme, an iron protein which was clearly involved in iron metabolism. They did this how? They did it by using iron deficient animals, iron deficient rats, and they looked for things that were upregulated in the gut, and they found this iron regulating gene, and they called it IREG1, iron regulating G1. Brilliant. They went to the meeting, which Paolo will remember very well, that was organised in Italy, in southern Italy. They talked about this, but they hadn't even written an abstract or they submitted anywhere. They talked about it openly, they explained the cloning strategy, the whole works. And to their great chagrin, before their paper was published, the following year a paper appeared from Nancy Andrews' lab using the zebrafish, in which she had used the same cloning strategy, but she was smart. She called the dump protein ferroportin. And now if you've got a choice between ferroportin and IREG1, I don't need to tell you what you're going to do. I spent the last edition of my book arguing that people should use IREG1, or at least quote it, because it had been the original. But their paper came out 4 weeks later. Don't talk about important findings. There is something called Febb's letters, which we'll publish it quickly. That's the way we publish the sequence of apophyrin when we were scared that anybody else might get out before us. it's the thing to do. Get something out in print before you go to a big meeting and talk about it. That was what you were talking about not talking openly at meetings about things that you haven't really got solid data. Right. Okay. So let's go through these dramatis personae that we're going to meet. The first one is transferrin. Now, transferrin is a diol protein. It's in fact made up of 4 lobes, but if you lay the 2 half sequences down one on top of the other, very homologous. It clearly came from an original duplication of a protein half this size. It's 80,000 molecular weight. Very big protein. And at each of these, you get one iron binding site here, one iron binding site. Now, this is, let me go for this. Does anybody know what this is?
DNA. DNA.
Yes. Well, give me its popular name.
Antithermal.
It's the Venus flytrap. It's the Venus flytrap. It's a carnivorous plant which is found in the Carolinas in the US, and it has very succulent-looking leaves. The poor, unfortunate insect comes down. It hits it, and that triggers something which makes the plant think, maybe something happening, but it doesn't act just yet. And if within the next 20 seconds, the unfortunate insect makes a second move and touches another of the hairs that are sticking up here, it just goes, boop, and that's it. End of story. Insect is slowly and quickly to be digested, and that's the way this protein works. There are a whole bunch of them. They are Venus flytrap proteins. They have an open conformation when they don't have the iron in. As soon as the iron comes in, the first couple of ligands bind, then the other 2 are brought in by the closing of this cavity, and you then have a nice closed structure. And there are lots of proteins that respond to this. So nature finds parallels, even at the molecular level, for things that we see at the much less molecular level. There is a transferrin receptor. And the transferrin receptor was discovered, would you believe it, by serendipity. That gives me the chance to tell my joke. Serendipity. Walter Grazzo, who used to write the molecular biology column for Nature, used to say that serendipity is not when you drop your butter toast and it falls butter side down. It does. It's when you pick it up and find the contact lens you lost last week. I went to a meeting and a German colleague from Bavaria came up to me afterwards and said, we have a better definition. I said, so what's your definition? He said, so when you're looking for a needle in a haystack and you find the farmer's daughter. Not quite so sophisticated, but anyway, you get the message. So serendipity, people were looking for a membrane protein that would be increased in cancer cells. And they kept finding this damn protein that was a disulfide bridge dimer, anti-kilodaltons, and it turned out it was transferrin receptor. Because of course the cancer cell wants to divide rapidly, it needs a lot of iron, it needs to increase the receptor that brings in the iron, so transferrin receptors are overexpressed in all cancer cells. Absolute standard. In fact, it makes the use of permanent cell lines very difficult for doing iron metabolism study, because you're then using cells that are overexpressing a protein that would normally not be expressed at anything like the levels that you get in a permanent cell line, field like cancer cells. So, the transferrin to cell cycle simply involves the dipheric protein binding to the receptor. It is then internalized into a clathrin protein vesicle. Clathrin protein vesicle is pumped full of protons, the pH down to about 5.5. At this pH, we understand that the iron comes out. It is reduced from iron 3 to iron 2 by this reductase. It's then taken out here by DMT1, which is an iron 2 transporter, a dimethyl metal transporter. The iron 2 stays in the cell. The transferrin remains bound to the receptor, is returned to the cell surface, and at the alkaline pH of the cell's pH 7.4 here, it has a low affinity for the receptor, and it dissociates and is then reused. So transferrin is recycled 100 or more times every time you're delivering iron. So we think that the mechanism is most likely that the transferrin receptor actually at pH 5.5 imposes the apo conformation on the diferric transferrin, which leads to the release of the iron, and then the equotransferrin hangs on until it comes back to the alkaline pH of the external. Ferritins are, and Paolo will tell you all about this later in the week, so I'm not going to even try. They are tetra 24 subunit proteins. They're heteropolymers of H and L. And when people talk to you about H-ferritin and L-ferritin, please do try to remember, in nature we don't have the capacity to clone and overexpress H-ferritin and L-ferritin separately. Human cells make a mixture of H and L. Some tissues have more H than L, some have more L than H, but it's always a heteropolymer. And the way in which the iron is incorporated seems to be pretty clearly that iron 2 is oxidized here by the H subunit, which is a peroxidase center. I told you that H-ferritins are part of the diiron proteins that we saw earlier. And then on the surface of the micelle, the iron is deposited. You can get down to about 4,500 iron atoms in a soluble, non-toxic and bioavailable form in ferritin. And ferritin can also aggregate, be internalized into lysosomes, where you then find it visible in electron microscopy in granules known as hemosiderin. I shan't tell you a story about a carload, a bootload full of starlings, because that would be, some people might not appreciate it. But when you go and go looking for starlings to isolate starling hemosiderin from liver, you need to get the birds first, and you need to bring them back. And of course, if they get out, you can get the problem. Anyway, let's move forward. Ferroreductases, we have several which are known, and one of these has been the structure of STEAP3 examined. This is the one that seems to be involved in the transfer of the cell cycle. DMT1 is a divalent metal transporter. This is a structure of a homolog from bacterium. And very recently, where did that go? Oh, this seems not to have been miraculous. Okay. Very recently, a ferroportin analog, I'll come back to ferroportin later. So, globally, in a human being, we absorb a milligram of iron a day from our diet, and we excrete a milligram of iron. If we excrete a little bit too much, we get iron loaded. The genetic condition known as idiopathic hemochromatosis is a well-known example of this. It's the most common genetic disorder. If we absorb too little, we become anemic. It's estimated that at least a third of the world's population is anemic. So you're on hiding to nothing. You're walking on a tightrope. A little bit too much one side, you get loaded. A little bit too much the other side, you're iron deficient. And we know, as far as the brain is concerned, I just remind you, that iron deficiency in neonates results in cognitive impairment, problems in learning. So iron is very important in early life for the brain. This gentleman said something which I should say in French, and I don't need to look up, I'll give you the translation. La stabilité du milieu interne est la condition d'une vie libre et indépendante. Claude Bernard, the father of experimental medicine. What he stated there is the essential principle of what we call homeostasis. It was said by Mr. Pickwick in one of Dickens' novels very clearly: income £1, expenditure 99 pence, state happiness. Income £1, expenditure £1, 1 pence, state misery. And possibly internment in a jail for netters. These were the days of Charles Dickens. He actually insisted that this was what we had to do. We had to maintain our internal medium at a constant level. And that means maintaining the level of iron within cells and within the whole organism at a level which is adequate, but not over the limit to be toxic, or not too little to be deficient. This is done at the cellular level by a very clever system of proteins that are called iron regulatory proteins. These proteins were discovered in Glasgow by Hamish Monroe. He was actually doing experiments on rats, and he saw that when you fed rats in their diet a large amount of iron, they laid down a lot of ferritin in the liver. And if you took the rats and you gave them actinomycin D, this is one of these classically simple experiments. Actinomycin D is a an agent which blocks RNA polymerase. The enzyme converts DNA to RNA, which makes messenger RNA. And if you added actinomycin D, the effect still took place. What this means is that there is a pool of messenger RNA sitting around there which is not being translated. And then subsequently, after we moved to Boston, he and others discovered that there are these proteins which we call iron regulatory proteins. And these iron regulatory proteins bind to regions in the ferritin case in the 5' untranslated region of the messenger RNA. Up here is the messenger, here's the message, here's the 5', here's the 3' untranslated region. In ferritin mRNA, they bind here when there's high iron, and the protein then, the translation of this is completely inhibited. Sorry, the other way around. Trying to go ahead of myself. In ferritin, when you've got lots of iron, this thing doesn't bind, so you get iron synthesized. But in the case of the transfer receptor, where you have the IREs in the 3' untranslated region, which is the part of the messenger RNA responsible for stability, the absence of binding means that this gets chopped up by nucleases, gets stabilized, and therefore you get synthesis of transfer receptors. So in high iron, you don't make the transfer receptor, you make the ferritin. When you've got low iron, exactly the opposite happens. Transfer receptor is upregulated because these proteins bind are protected. Ferritin is not synthesized. So you've got a nice yin-yang mechanism where if iron's in too great amount, you increase the storage protein and you decrease the uptake system. And if you've got too little iron, you do that. That's at the cellular level. Now, at the whole organism level, and this is a slightly, you'll see this kind of thing in a much more sophisticated version. This is how it begins to look when you start adding in all the twiddly bits. I've left most of the twiddly bits out so you can understand it. I made a mess of explaining. Right, I had to put this slide up for a very simple reason. When you publish a book, as I've done, this small tome here, you have to get permission to reproduce all of the figures. And the original paper in which hepcidin, that structure there, the corrected hepcidin structure is shown, is published in JBC. JBC, I think it's $245 a figure, something like that. They want to charge you to reproduce the figure. So, actually, the figure that's in the book of hepcidin is from camel hepcidin. Not because I like camels, but because that's one that was published in a journal that allows you free publication. But my publisher decided to put it on the cover of the 4th edition of my book. So here it is. And they were prepared, they were able to negotiate and get permission to reproduce this from JBC. So, well done there. That was more than I could afford. But it's actually that, when it's the author who pays the fee for reproduction out of his royalties. So it makes writing a book hardly worthwhile. So, the way in fact hepcidin works is it acts on ferroportin. We saw that ferroportin is an iron export protein, and it's in the membranes of iron exporting cells. But when hepcidin binds, it internalizes this and degrades it. And so you stop iron export from the cell, and you just build up iron inside the cell. This is a very simple version that I'm looking down, I published a few years ago, which just shows you that the liver produces hepcidin. Hepcidin goes to block iron release from the iron stores and the macrophages of the spleen. It blocks iron release from the duodenum, so it blocks iron absorption from the gut. And it also affects expression of hepcidin. I'm not going to go into great detail of hepcidin. Hepcidin is very important in the regulation of systemic iron homeostasis, as is erythropoiesis and hypoxia. And it all gets much more complicated, as you can see in these slides. And the cascades are not even in there. Once you start putting the amplification cascades in, it becomes a nightmare. I'm going to go to the end of the talk. You'll be very glad to hear I'm actually going to finish more or less on time. Oxidative stress can be caused by iron, and certainly one of the major sources of oxidative stress, as I said earlier, is the hydroxyl radical. But you also can get nitrous oxide, which is quite often used as a signal. You can get peroxynitrite. You get these radicals over here, and you can get chloride. These are the nasties. You defend yourself from these nasties in a battlefield where you use a series of enzymes, which are glutathione peroxidase, glutathione reductase, superoxide dismutase, catalase, peroxidase, and try to protect yourself with vitamins, vitamin C, with vitamin A. So this is what's all going on in your body all the time. What we know is that one of the major defenders is glutathione, one of the best things against oxidative stress. And glutathione is relatively low in concentration in the brain. So the brain is probably much more susceptible to oxidative stress than the rest of the body. And here I end up with a slide to illustrate what happens when you have transferrin. Usually when it's circulating is only about 30% saturated. But when you get iron overload, you can get iron saturation going up and up. And as iron saturation gets above 60 or 70%, you start to see a form of toxic iron, which is called non-transferrin bound iron. Non-transferrin bound iron is essentially taken up by hepatocytes and is extremely damaging in terms of its toxicity. Now, there are indications in the literature that the cerebrospinal fluid, which contains transferrin, the transferrin in there is probably very highly saturated, suggesting that there may well be non-transferrin bound iron in the interstitial fluid and the CSF of the brain. So this may explain some of the strange anomalies in iron uptake by brain cells, which you may be hearing about over the next few days. The other thing which is troubling is that, of course, oxidative stress is also associated with the aging process. And so I want to end my talk with this beautiful picture by Titian, which hangs in the National Gallery of Scotland, and which is called, appropriately, the three ages, not of civilization, but the three ages of man. From the young child, to the young lovers, to the elderly gentleman contemplating a skull. And this is just reminding you that neurodegenerative diseases, of which problems of myelination are but one, are frequently associated with the aging process. They are much more frequent in elderly patients. We have a system of care in Europe which has been up till now fairly good at handling patients, as long as we don't get too many elderly patients in hospital at one time. But as we begin to get more and more elderly patients, we begin to find that we don't have the social means to look after them at home. And so we get the crazy situation where beds are no longer available for patients coming into accident and emergency units. Patients are lying around on trolleys and waiting outside the hospital in ambulances because there are no beds available, because the available beds are occupied by elderly patients who are prepared to go home, but for whom the social care facilities don't exist. This is a social problem. It's not just a trivial problem, and it's also related to neurodegeneration. Because if we could start picking up some of these neurodegenerative diseases earlier, we might be in a better position to make sure people can be looked after better at home rather than having to be hospitalized. That's just the end of the story. I'd like to thank you for your attention. I wish you a very good afternoon. Thank you.